So xylem conveys water and dissolved minerals upward from the roots into the shoots. Phloem transports organic nutrients, sugars, from where they are made by photosynthesis in the leaves to where they are needed in the plant for growth. The vascular system of a stem or root is together called the stele, S-T-E-L-E. In angiosperms or flowering plants, the stele of the root is a central vascular cylinder. And then the stele of stems and leaves is divided into vascular bundles, strands of xylem, water, and flower's food. So tissues that are neither dermal nor vascular are the ground tissue system. The ground tissue includes cells specialized for storage, photosynthesis, and so there are different types of plant cells, and these are illustrated on page 744. There's parenchyma, collenchyma, sclerenchyma, water-conducting cells of the xylem, and sugar-conducting phloem. So up here is parenchyma. These you can see with the light microscope. This is an Elodea leaf, and these are chloroplast. Okay, and you can see the cell walls. So they have primary cell walls that are thin and flexible, and most lack secondary walls. Down below here is collenchyma cells, and these are grouped in strands or cylinders, help support young parts of the plant. And they have thicker primary walls than the parenchyma cells up above, though the walls are kind of unevenly thickened. Young stems and petioles often have strands of collenchyma cells just below the epidermis, okay? So this is what the collenchyma cells cause the strands in a celery stalk, okay? The strings of a celery stalk, which is a petiole. Okay, down here, these are sclerenchyma cells, and these are strengthened by the lignin and are much more rigid, okay? Just much more rigid. Then over here we have water-conducting cells of the xylem, okay? These tracheids are found in xylem, and most angiosperms, as well as some gymnosperms, have these vessel elements. So when the living cellular contents of a tracheid or vessel element disintegrate, the cell's thickened walls remain behind, forming a non-living conduit through which water can flow. Okay, so xylem does not have to be alive. Sugar-conducting cells of the phloem. So, unlike the water-conducting cells of the xylem, the sugar-conducting cells of the phloem are alive at functional maturity. So that's a big difference. Remember that. In seedless vascular plants and gymnosperms, sugars and other nutrients are transported through long, narrow cells called sieve cells or sieve cells. In the phloem of angiosperms, these are sieve tubes. So whenever you see the word sieve, you know that they're talking about phloem. Okay, apical means tip. Meristems means active growth. So the apical meristems are located at the tips of the roots and in the buds of shoots. If you remember back to your mitosis lab in BIO-101, you looked at apical meristems of the roots of onions, okay, which were actively growing, and you saw active mitosis, so actively growing. And apical meristems elongate shoots and roots in a process called primary growth. Now, that's primary growth. Okay, lateral meristems add thickness, okay, to woody plants, a process called secondary growth. And there are two lateral meristems, the vascular cambium and the cork cambium, okay? So the vascular cambium adds layers of vascular tissue called secondary xylem or wood and secondary phloem. The cork cambium replaces the epidermis with periderm, which is thicker and tougher. So these two cambiums add to secondary growth. Okay, so here are apical meristems, vascular cambium, and then here, cork cambium, lateral meristems. Here are root apical meristems. So here's top and bottom, primary growth. This is secondary growth. So primary growth in stems, you have the epidermis, the cortex, metabolic function, primary phloem, primary xylem, and the pith. So this is at the top, okay, new growth. This is older growth down here. Secondary growth in stems, you have periderm, cork cambium, cortex, primary phloem, secondary phloem. Here's the vascular cambium, secondary xylem, primary xylem, and pith. So the xylem, primary and secondary xylem, this is what adds rings of gray inside of the plant. So in woody plants, primary and secondary growth occur simultaneously, but in different locations. Okay? So down here, the growth of 2 years ago, 3 years old. Here's a leaf scar. So this is a little bigger. Scars left by terminal bud scales of previous winters. Okay, these have fallen off. Last year's growth, 2 years old. Not as thick, not as much secondary xylem. And then this 1 year side branch formed from an axillary bud. Okay, here are nodes. Here's the inner node. And then this year's growth. Here's the stem, leaf scar here, auxiliary buds, bud scale, and terminal bud. So if you would prune, like if you want to make this bushier, you prune right above this leaf scar, and then this will branch here. This might have been pruned here to make this branch. So pruning makes a plant full. So primary growth produces the primary plant body, the parts of the root and shoot systems produced by apical meristems at the root and up. So primary growth of roots. The root tip is covered by a root cap, okay, which protects the apical meristem because that root has to push through soil. Then just behind the root tip are 3 zones of cells, okay? Zone of division, rapid mitosis, zone of elongation, and zone of maturation, okay, where differentiation occurs. So here is the root cap, protects this apical meristem here. Here a lot of mitosis is going on here. Here it's gaining growth, zone of elongation. And here is zone of maturation where root hairs extend, and this increases absorption area. So here's the vascular cylinder, here's the cortex, and the outer coating, protective coating of the epidermis. So the primary growth of roots produces the epidermis, the ground tissue, and vascular tissue, just like I pointed out in the previous slide. And in most roots, the stele is that center vascular cylinder. The ground tissue fills the cortex, which is between the vascular cylinder and the outer coating or epidermis. So the innermost layer of the cortex is called the endodermis. Okay, so here's the primary growth of roots produces the epidermis, ground tissue, and vascular tissue. And you'll be looking at some of these slides in the lab. So this is a typical root of gymnosperms or eudicots, and you can see the vascular cylinder here, cortex, epidermis. Okay? So let's take a closer look at this vascular cylinder, and you can see how it's divided. These round, large spaces are xylem, transporting water. Around it, within this vascular cylinder, is the phloem, transporting food. Okay, over here, this isn't a monocot root. Okay, so it's different. We've got epidermis, cortex. This is the vascular cylinder. This is the endodermis, okay, which we mentioned before, pericycle. And here, these elements here are xylem, and then around here are phloem. Okay, so these two cross sections of roots look very different between monocots and dicots. Okay, and lateral roots arise from within the pericycle, the outermost cell layer in the vascular cylinder. So the vascular cylinder in the middle. So here's the vascular cylinder, cortex, that's the epidermis. Here's an emerging lateral root. And you can see, as time goes on, the lateral root emerging, with the root cap, the meristem, the area of division, rapid growth, elongation, and then maturation back. Okay, so that was roots. This is the primary growth of shoots. So a shoot apical meristem is a dome-shaped mass here of dividing cells at the tip of the terminal bud. Okay, so a lot of mitosis here. It gives rise to a repetition of internodes and leaf-bearing nodes. Okay, so here are axial bud meristems. Here is developing vascular strands. Leaf primordia, these are the beginnings of leaves. Okay. Tissue organization of stems. So in gymnosperms, like conifers, most eudicots that have branching veins in their leaves, the vascular tissue consists of vascular bundles that are arranged in a ring. So in most monocot stems, they have parallel veins like corn, grasses, a lot of bulbs like daffodils, tulips. The vascular bundles are scattered throughout the ground tissue rather than a ring. Okay, so here you can see a ring is formed. This is more advanced plants, a eudicot, flowering sunflower. Vascular bundles form a ring. Okay. Ground tissue toward the inside is called pith, and ground tissue towards the outside is called the cortex. And here are vascular bundles. Okay. This is phloem, and these round little structures are xylem. Okay. And then epidermis on the outside. Okay, so here's a monocot, a maize or corn stem. Vascular bundles are arranged throughout. Here's the ground tissue, but these are xylem here and phloem. Vascular bundles, whenever you see the word vascular, you know it's in phloem. So the epidermis in leaves is interrupted by stomata, and I hope you remember what that means. They have guard cells on each side, and they regulate the passage of CO2 exchange between the air and the photosynthetic cells in a leaf, and oxygen also leaves that through the stomata, and also water with transpiration. So the ground tissue in a leaf is sandwiched between the upper and lower epidermis, and the vascular tissue of each leaf is continuous with the vascular tissue of the stem. That's what gives the leaves its veins.